let's talk a little bit in more depth about how DNA actually copies itself, how it actually replicates. And we're going to talk about the actual actors in the process. Now, as I talk about it, I'm going to talk a lot about the 3' and the 5' ends of a DNA molecule. And if that is completely unfamiliar to you, I encourage you to watch the video on the anti-parallel structure of DNA. And I'll give a little bit of a quick review here just in case you saw it, but it was a little while ago. This is a zoom in of DNA. It's actually the zoom in from that video. And when we talk about the 5' and 3' ends, we're referring to what's happening on the riboses that form part of this phosphate sugar backbone. So we have ribose right over here, 5 carbon sugar, and we can number the carbons. This is the 1' carbon, that's the 2' carbon, that's the 3' carbon, that's the 4' carbon, and that's the 5' carbon. So this side of the ladder, you could say, it is going, let me draw a little line here. This is going in the 3' to 5' direction. So this end is 3', and then this end is 5'. It's going 3' to 5'. Notice this phosphate connects to the 3', then we go to the 5', connects to a phosphate. This connects to a 3', then it connects, and then we go to the 5', connects to a phosphate. Now on this end, as we said, it's anti-parallel. It's parallel, but it's oriented the other way. So this is the 3', this is the 5', this is the 3', this is the 5'. And so this is just what we're talking about when we talk about the anti-parallel structure. These two backbones, these two strands are parallel to each other, but they're oriented in opposite directions. So this is the 3' end, and this is the 5' end. And this is going to be really important for understanding replication because the DNA polymerase, the things that's adding more and more nucleotides to grow a DNA strand, it can only add nucleotides on the 3' end. So if we were talking about this right over here, we would only be able to add going that way. We wouldn't be able to add going that way. So one way to think about it is you can only add nucleotides on the 3' end, or you can only extend DNA going from 5' to 3'. If you're only adding on the 3' end, then you're going from the 5' to the 3' direction. You can't go from the 3' to the 5' direction. You can't continue to add on the 5' side using polymerase. So what am I talking about with polymerase? Well, let's look at this diagram right over here that really gives us an overview of all of the different actors. So here is just our DNA strand, and we could imagine it's just somewhat natural in its natural unreplicated form. And you could see we've labeled here the 3' and the 5' ends, and you could follow one of these backbones. This 3', if you follow it all the way over here, it goes, this is the corresponding 5' end. So this and this are the same strand, and this one, if you follow it along, if you go all the way over here, is the same strand. So this is the 3' end of it, and then this is the 5' end of it. Now the first thing, we've talked about this in previous videos where we gave an overview of replication, is the general idea is that the two sides of our helix, the two DNA, the double helix needs to get split, and then we can build another side of the ladder on each of those two split ends. You could really view this as if this is a zipper, you unzip it and then you put new zippers on either end. But there's a lot of, in reality, it is far more complex than just saying, oh, let's open a zipper and put new zippers on it. It involves a whole bunch of enzymes and all sorts of things. And even in this diagram, we're not showing all of the different actors, but we're showing you the primary actors, at least the ones that you will hear discussed when people talk about DNA replication. So the first thing that needs to happen right over here, it's all tightly wound. So let me write that. It is tightly wound. And it actually turns out the more that we unwind it on one side, the more tightly wound it gets on this side. So in order for us to unzip the zipper, we need to have an enzyme that helps us unwind this tightly wound helix. And that enzyme is the topoisomerase. And the way that it actually works is it breaks up parts of the backbones temporarily so that it can unwind and then they get back together. But the general high-level idea is it unwinds it, so then the helicase enzyme, and the helicase really doesn't look like this little triangle that's cutting things. These things are actually far more fascinating if you were actually see the molecular structure of helicase. But what helicase is doing is it's breaking those hydrogen bonds between our nitrogenous bases. In this case, this is an adenine here, this is a thymine. It would break that hydrogen bond between these two. So first you unwind it, then the helicase, the topoisomerase unwinds it, then the helicase breaks them up, and then we actually think about these two strands differently. Because as I mentioned, you can only add nucleotides going from the 5' to the 3' direction. So this strand on the bottom right over here, which we will call our leading strand, this one actually has it pretty straightforward. Remember, this is the 5' end right over here, so it can add going in that direction. It can add going in that direction right over here. This is the 5' to 3'. So what needs to happen here is, to start the process, you need an RNA primer, and the character that puts an RNA primer, that is DNA primase. We'll talk a little bit more about these characters up here on the lagging strand, but they'll add an RNA, let me do this in a color you can see. An RNA primer will be added here, and then once there's a primer, then DNA polymerase can just start adding nucleotides. It can start adding nucleotides at the 3' end. And the reason why the leading strand has it pretty easy is this DNA polymerase right over here, this polymerase, and once again, they aren't these perfect rectangles as on this diagram. They're actually much more fascinating than that. You see a DNA polymerase up there. You also see one over here, polymerase. This polymerase can just, you can kind of think of it as following the open zipper and then just keep adding nucleotides at the 3' end. And so this one seems pretty straightforward. Now, you might say, well, wouldn't it be easy if we could just add nucleotides at a 5' end? Because then we could say, look, this is going from 3' to 5'. Well, maybe that polymerase or different polymerase could just keep adding nucleotides like that, and then everything would be easy. Well, it turns out that that is not the case. You cannot add nucleotides at the 5' end. And let me be clear. This 3' right over here, this I'm talking about this strand. This strand over here, let me do this in another color. This strand right over here, this is the 3' end, this is the 5' end. And so you can't just keep adding nucleotides just like that. And so how does biology handle this? Well, it handles this by adding primers right as this opening happens. It'll add primers, and this diagram shows a primer is just one nucleotide, but a primer is typically several nucleotides, roughly 10 nucleotides. So it'll add roughly 10 RNA nucleotides right over here, and that's done by the DNA primase. So the DNA primase is going along, the lagging is going along this side, you could say the top strand, and it's adding the RNA primer, which won't be just one nucleotide, it tends to be several of them. And then once you have that RNA primer, then the polymerase can add in the 5' to 3' direction. It can add on the 3' end. So then it can just start adding DNA like that. And so you can imagine this process, it kind of, you add the primase, put some primer here, and then you start building from the 5' to the 3' direction. You start building just like that, and then you skip a little bit, and then that happens again. So you end up with all these fragments of DNA, and those fragments are called Okazaki fragments. So the Okazaki fragments. And so what you have happening here on the lagging strand, you can think of it as, why is it called the lagging strand? Well, you have to do it in this kind of, it feels like a suboptimal way, where you have to keep creating these Okazaki fragments as you follow this opening. And so it lags. It's going to be a slower process. But then all of these strands can be put together using the DNA ligase. The DNA ligase. Not only will the strands be put together, But then you also have the RNA being actually replaced with DNA, and then when all is said and done, you are going to have a strand of DNA being replicated or being created right up here. And so when it's all done, you're going to have two double strands, one up here on the lagging strand and one down here on the leading strand.